Hello guys and welcome to another lesson with Nogops. Today we are going to talk about DNA replication and mitosis, and I'll have you guys know that mitosis, I mess with mitosis. Alright, mitosis is my thing. We mess with mitosis, so this one's going to be a good one. Alright, Nogops, you're in good hands. So we, as usual, are just going to give Nogops her glasses, really get her ready for what we've got today. So, before we get into mitosis, we need to talk about DNA replication. So this is very different than transcription. Transcription is, you've got DNA, which is the recipe book, and we're taking a recipe from it. Replication is we are copying the whole recipe book. So it's quite different. But before we start, we're just going to talk about parent cells. So you got parent cells, and those parent cells will divide to form daughter cells. So the parent cell is going to give you two daughter cells. Daughter cell and then daughter cell. Frick. Hold on, I thought I was smart with that. Alright, whoa. Daughter cell. Well, daughter cell. And these are going to be identical cells with the same DNA. Alright? Same DNA as the parent cell, and they both also have the same DNA. That's very important. They have the same DNA. In multicellular organisms, the new daughter cells, they are going to become part of the existing organism. But in unicellular organisms, the daughter cells are going to form a whole new organism. But we are going to be talking about multicellular, so we don't have to worry about that. So, there are some things called germ cells. They form from two parents, but those are created by meiosis. Meiosis. We're going to talk about meiosis later. Mitosis is asexual reproduction, and meiosis is sexual cellular reproduction. So, basically, just to cover that a little bit, germ cells, which are egg and sperm cell, they have different DNA than the parent cell. So this is where mitosis and meiosis are different. Mitosis creates daughter cells that have the exact same DNA as the parent cell, whereas meiosis creates germ cells that have a different DNA than the parent cells. Because the germ cells, what they do is they combine DNA from one parent and the other parent, and they mix them together to create this unique genetic makeup. And this is called a zygote. We will cover that in the meiosis lesson. Today we're talking about mitosis. So before a cell reproduces, before the cell division happens, it needs to replicate its DNA. We need to replicate this DNA. So that's where something called a chromosome comes into play. So a chromosome is just DNA containing structures. They are in the nucleus, and they have DNA segments called genes. So each chromosome is a strand of chromatin. Chromatin is just DNA and protein. And these protein molecules are really important because they regulate the expression of genes, and they keep the chromatin organized. So we're going to draw like a little example of the DNA, and you'll see like the different types of DNA. So the first type of DNA, this is going to be unpacked naked DNA. We're just going to write naked DNA. This is a freaky DNA. Naked DNA, and it looks something like, remember how we said it's a double helix? So it looks something like this. Imagine this is like twisting. Okay, it's kind of hard to draw twisting. Alright, this is naked DNA. It's just no proteins attached. It's just unpacked, naked. And this is used for transcription, which you should know, Nogops. We talked about that. And DNA replication. So this is good for us. Alright. Now, if we keep moving down the DNA, hold on. Keep moving down the DNA. Alright. It will start looking something like this. Let's get a different color. Let's get a purple up in here. Actually, we'll do green. All right, so there's gonna be these little circles in the DNA, and these are gonna be like wrapped around. So let's, how do I draw this one? I'm not artistic in this format. So there's gonna be these little bundles. All right, and that's the protein wrapping around. And let's see, it wraps, and then we have another bundle here of proteins. So the green is the protein, and then the blue is just the DNA wrapping around the protein. So do you see how when I mentioned that the proteins, they help regulate the expression of genes and they keep the chromatin organized? So this is the chromatin being organized. So this right here is uncondensed chromatin. So let's do this. This section is this. This section is gonna be second form of DNA, which is uncondensed chromatin. All right, so when it's wrapped up like this, it's uncondensed chromatin. And so this right here goes all throughout the entire DNA, and it's like a big, big strand. And eventually it looks like very, very condensed. I'm gonna change the color to really show you how condensed it is. Very condensed and thick line. Hold on, let's make a thick. Very condensed, thick line. And this right here, this thick and condensed line, this is the condensed chromatin. Whoops. Condensed chromatin. And one thing before we leave off at the uncondensed chromatin, this is DNA wrapped as a nucleosome. So a nucleosome is one protein, it's DNA plus 8 histone proteins. So the proteins that are in DNA are histone proteins. So basically one little circle. Well, I guess I misled it there because I put... No, I did it right. Look at me go. So one little nucleosome has 8 histones in there. So it's DNA plus 8 histones. We got 8 in there. So that is uncondensed chromatin. And when the chromatin is uncondensed, individual genes can be unpacked for transcription. And that's why it's important for the chromatin to be uncondensed. Condensed chromatin is when the chromatin coils, loops, and it folds really tightly, creating this like thick chromatin. And this is when the chromosome becomes visible in the nucleus because it's so thick. So that's important to know, is that chromosomes become visible in the nucleus. I don't care about spelling. Don't hold me on that. So chromatin must be condensed during cell division so chromosomes can be separated. All right, so that has to happen. Otherwise, we cannot separate the chromosomes. So, but condensed chromatin cannot be used for transcription and DNA replication. So that's also something to keep in mind. So just a little review of what we just did. When an egg and a sperm combine, they form a zygote, which has a unique genetic makeup. Mitosis is used... Oh, I forgot to mention. Mitosis is used for growth and repair of tissues. All right? Growth and repair of tissues, while meiosis is obviously to make a whole new human being and make a whole new organism. So, a chromosome is a single strand of DNA and its associated proteins. 8 histone proteins come together and they form a strand of DNA. So these are 8 histone proteins right here, and they form... they don't form a strand of DNA, they form a nucleosome. DNA has to be unpacked and uncondensed in order to be used for DNA replication. And chromatin is the collection of DNA and protein contained in the nucleus. All right, we're good on that. We're good on that. So now we're gonna talk about DNA replication. So DNA replication, pretty similar to, what is it called? Transcription, in terms of like separating the helicase and all that fun stuff. So let's bring Nogops down here. So we have our helicase. All right, and we've got our nitrogenous bases right here. Whoops, just a bunch of nitrogenous bases. All right, so the DNA helicase will come and it will unzip the DNA helix, and this will reveal the nitrogenous bases. So these red things are the nitrogenous bases. So basically, the DNA helicase, it's like an enzyme, and it comes right here, and it separates these. It's like moving them apart, so this gets separated from this. And then there are things called origins of replication. They're going to be located in the cell, in the DNA too. So that's where the DNA polymerase comes in, and it starts using both the DNA strands to make the complementary daughter strand. So you're going to have like a little DNA polymerase right here, and it's going to be making a new strand. Let's do pink. A new daughter strand by connecting it and like forming bonds with it. So at the end, after it's done forming bonds, you're going to have a daughter strands that are a copy of the DNA. So all that happens is the DNA helicase separates these, and then the what is it called? The DNA polymerase. Let's do a different color. Let's say this is the DNA polymerase. It starts forming hydrogen bonds like here, until it makes a duplicate of the DNA. And so if the DNA, using the codes again, nitrogenous bases, if the DNA sequence was A, T, G, A, C, C, T, then the complementary daughter DNA sequence would be T, A, C, T, G, G, A. It's just the complementary bases, but we're only using the DNA bases. Unlike transcription, we were using the RNA bases, where we swap the T for a U. Where we don't use T and we use U instead. This one is just the DNA bases, just as it is. And one more thing, the enzyme that positions the new complementary nucleotides along like the template strands and what basically makes this is called ligase. It joins the daughter strand nucleotides with covalent bonds. This process is very similar to transcription, except instead of a RNA polymerase, we're basically using a DNA polymerase. That's why I'm going by very quick. Are you good on that, Nogops? So let's get into the fun part. Actually, no, not yet. One more thing about chromosomes. So let's do it over here. So, DNA replication. We'll take this is our chromosome. Alright, she kind of just looks like this. So this is a chromosome. Let's say one chromosome. Okay, that's one chromosome. When you do DNA replication, you will get two of these strands. Alright, you'll get two of these strands, and those strands, they're going to be held by something right here. This is called a centromere. So after replication, you will have two strands and a centromere. These two strands are called sister chromatids. These are sister chromatids. So you go from one chromosome to two sister chromatid. But this is still one chromosome. All right? So you still have one chromosome. Just because you have two strands of chromatin does not mean you have two chromosomes. That's a very important detail. This is one chromosome. All right, one chromosome, not two chromosomes. It has two sister chromatids, but only one chromosome. All right, now we're going to go into the cell cycle, aka cell division. I love cell division. So let's grab Nogobs with us. And we need a lot of space for this. So we're gonna pop you right here. Alrighty. So we have phases. Let me draw it how they drew it in the book, because they drew it pretty nicely. Let's do a circle. Alright, that's the cycles. We have G1 phase. Alright, then we have S phase. Alrighty, and then we have G2. And then we have M phase. Alright, and then M phase has different phases. Alright, like this. And then this one in the middle, I phase. And we move in this direction. Alrighty, perfect. So, we're going to start with our first phase, which is interphase, the brown. Interphase is the phase where the cell carries out its normal cell functions of growth and metabolism. So this is just a normal cell. It's doing its normal cell things, and this occupies most of the cell's time. So most of your cells in your body are doing this right now. And this interphase consists of 3 subphases. What are those 3 subphases? Well, it's G1. Let's start with G1. So G1 is where the cell grows and gathers material for DNA replication. So this is DNA replication prep. So I'm going to say prep for DNA replication. All right, so it's gathering materials, it's growing, organelles duplicate, and the cytoplasm volume is increased. Okay? And then S phase, what color did we do S? Orange. S phase is when the DNA is actually replicated. So DNA is replicated. So that's where what we talked about up there, where the DNA helicase unzips and then the DNA polymerase comes in and it creates those sister chromatids, and it creates double-stranded chromosomes. All right, double-stranded chromosomes does not mean double the chromosomes. So now we have the last phase before mitosis, and that is G2. And in G2, the sister chromatids are joined by a centromere, appears, and then cell prepares for division. So this consists of checks and repairs to the chromosome. So we're going to do check, repair, chromosome. So these 3 steps need to be done before cell division can even start. So G1, we prep for DNA replication, we gather material, organelles duplicate, and the cytoplasm volume increases. S phase is where the DNA is actually replicated, and we create double-stranded chromosomes. So we go from this one single-stranded chromosome to one double-stranded chromosome. And then in G2 phase, the centromere appears and it connects the sister chromatids together. And it also, in G2 phase, we check and repair the chromosome for any damage or any errors in the duplication process. Now we move into something called the mitotic phase, or the M phase. This is where the drama happens, where everything goes down in the mitotic phase. So this is the phase where the cell divides into two daughter cells. And this involves two main tasks. So the two main tasks for this are divide the nucleus and chromosomes. And this takes place in the P, M, A, and T phases. And then I'm gatekeeping them for now. You guys don't get to know what they mean. And then we have the second job, which is divide the cytoplasm and organelles. Alright, and this happens in a process called, let's change the color, cytokinesis. Alright, cytokinesis. So now let's actually get into it. So we went over interphase. We got G1, prep for DNA replication, gather materials, organelles duplicate, and cytoplasm volumes increased. S phase, DNA is replicated, creating double-stranded chromosomes. G2 phase, sister chromatids are joined by a centromere, and the cell prepares for cell division by checking for any errors in the chromosomes. And then we move on to the mitotic phase. In the mitotic phase, there's two responsibilities: divide the nucleus and the chromosomes, and then divide the cytoplasm and organelles. So let's get into the mitotic phase. What happens in the mitotic phase? So we are going to go back to interphase to cover this, but let's grab Nogops first and bring her down. Once again, we're going to have to get pretty artsy with drawing, so don't worry. You're in good hands. So we're going to start once again with interphase. Our first ever phase is interphase. So what does the cell look like in interphase? The cell looks like this. And then we have this. And then let's draw these little things right here. Alright. And then inside of the cell, let me get a purple color. You have like the chromatin. Alright. So we already know that in interphase there's S phase, G1, G2. So in S phase is when the DNA is replicated. Alright. And one important thing to note here is that the chromatin is uncondensed. Uncondensed chromatin. So that's why it looks like squiggly lines, because it's uncondensed. No, not unconnected. Uncondensed chromatin. And then this right here, these little orange things, these are centrioles, and they form. So we're going to put that down. Centrioles form at the end of G2. So at the end of G2 phase, these centrioles form, and they become visible. And they are formed outside of the nucleus. So now we get into the real stuff. Now, our next phase is prophase. Prophase. And in prophase, we're going to divide it into two things. We're going to divide it into, let's see what color, let's do green. Early prophase and then late prophase. But let's start with early prophase. So in early prophase, the cell is going to look very different. It's going to be like this. And then let's do our nucleus. But now, see now, the chromatin condensed. It starts to condense and it becomes condensed chromatin. So if you remember from our little diagram up here, we just went from this to this. So the chromatin becomes condensed, and then these little things that were up here, they start to slowly go to the opposite ends of the cell, and they start forming things called mitotic spindles. Mitotic spindles. Alright, so the centrioles, they start forming mitotic spindles, and the chromatin starts to condense. Alright, and the orange, the centrioles, they move to the opposite sides of the cell, and they grow mitotic spindles. So early prophase, we're going to say chromatin condenses, and centrioles move to opposite sides, and finally mitotic spindles form. Those form. Alright. One last thing that happens in early prophase is the nucleus is actually going to disappear. It's going to start disappearing. Sorry, the nucleolus is going to disappear, not the nucleus. This, whoa, appeared. Alrighty, that's early prophase. Now, let's see, what the hell happens in late prophase? Late prophase. So in late prophase, the nuclear envelope is going to disappear completely. So we are going to have a cell like this, and we are going to have our centrioles. Remember, they went to the opposite sides, and they have their mitotic spindles out. They're ready to grab. Just like this. And then our chromatin, condensed chromatin, we're going to call it chromosomes, they are going to attach to these things. They're just going to randomly attach. Alright, and we have a lot of these chromosomes, alright? And the the mitotic spindles are going to attach to these centromeres on the chromosomes. So let's attach them. These are going to attach, to the centromeres on the chromosomes, and they're going to start to slightly pull on them. Alright, so what happens in late prophase? Well, first of all, the nuclear envelope disappears, and the chromosomes are freed. And then the chromosomes attach to the mitotic spindles. I'm just going to say spindles, I don't want to write the whole thing. And then the centrioles, using the mitotic spindles, they are going to pull on the chromosomes. And they're going to pull them towards the middle. Alright? Because we need the chromosomes in the middle. So, in prophase, in early prophase, we're going to see the chromatin condense. We're going to see the centrioles move to the opposite sides of the cell. We're going to see these mitotic spindles form, and the nucleolus will disappear. In late prophase, the nuclear envelope disappears, so this purple envelope disappears. Chromatin are free. The chromosomes are going to attach to the mitotic spindles. The centrioles are going to pull the chromosomes towards the middle. That leads us to our next phase, which is metaphase. So let's bring Nogops down here to really show her what's up in metaphase. Alright. Let's do metaphase in orange. Yeah. Metaphase. Alrighty. In metaphase, the cell is gonna look like this. Alright, we got our centrioles right here. Alright, and then we got our mitotic spindles coming out of there. Actually, might need to make these longer. Alright, and our chromosomes, they are gonna line up in the middle of the cell. So remember how before we said that the mitotic spindles are gonna pull them to the middle? That's what they're gonna do. They're gonna pull them to the middle, and they're gonna line them up for metaphase. So the centrioles continue pulling on either side of the chromosome centromere, and the microtubules are gonna pull on the centromeres. That's literally all that happens in metaphase, is we just line up the chromosomes in the middle. So think of metaphase as met, like, you know, they meet. They met each other. They meeting in the middle. Like, why don't you just meet me in the middle? That's pretty much what's happening in metaphase, and that's the only thing that's happening in metaphase. So the next phase is going to be anaphase. Have we used pink? No. Anaphase. Ooh. Nogops got a little excited for anaphase. I love anaphase. Who does not like anaphase? Alright. In anaphase, this is where it gets real. The cell looks like this. We got our centrioles, our mitotic spindles. Whoa. Alrighty, and they pulled them completely apart. So now you have like this. They are pulled apart. They pulled the chromosomes apart. So now you have single-stranded daughter chromosomes. So the chromatids, the sister chromatids, they're going to get split in half and separate into single-stranded daughter chromosome. Alright? Now, let's just write that down first. So, no, let's do sister chromatids pulled apart. So the sister chromatids get pulled apart, and that leaves us with single-stranded daughter chromosomes. Now remember up here when I said that this is one chromosome and this is also one chromosome? So what do you think happens when this separates? When this separates, we get, here, let's separate them. All right, when that separates, we get 2 chromosomes. All right, this is the confusing part, all right? So this is one chromosome. When they are joined together, when they are connected together with the centromere, it is still one chromosome. But when it's one strand and one strand that are not joined together, it is 2 chromosomes, all right? So how does this apply? These two separated, right? We had these. And now they separated. So that means, this is a very important piece of information. That means that the chromosome number in the cell is doubled. So now we have double the chromosomes. remember? Because they separated. So now it's doubled. That is really important. So anaphase is the phase in the cell where the chromosome number is doubled, all right? So when they get doubled, they get pulled. The microtubules, they pull them, they shorten, and they pull the chromosomes towards the cell poles. So these are the cell poles. And then we go on to, one last thing. The microtubules, this pulling and pushing is going to naturally elongate the cell into a more oval shape. And this leads us to our final phase. Let's move her down. Our final phase, let's do our final phase in red, is telophase. What happens in telophase? Hold on, let's make this look nicer. Telophase, the cell is going to look like this. So all right. And then let's get our DNA up in here. No, actually, let's just stick to these strands of DNA. And then we have, can't forget these right here. And then let's use this. Alrighty. So this is where the cell starts to really separate. So the daughter chromosomes, they reach the cell poles. So these are the daughter chromosomes. They reach the cell poles at the centrosome and they uncondense into chromatin. All right. So they become squiggly lines again. They become uncondensed. Here, actually, yeah, I kind of drew it confusing. So they become squiggles again. They uncondense. So that's really important to know that they uncondense. Chromatin uncondenses. All right. And after they uncondense, the nuclear envelope reforms. So they are back. The nucleolus is back. So and they form around each of the new daughter nucleuses. So now we have two nucleuses because we technically have two cells. And then this little indentation right here, this indentation, this is the cleavage furrow forming. It is an indent that forms due to the contracting ring of the microfilaments to pinch the cell into two. So that's what this base stuff is. These are microfilaments. They're pinching the cell into two, and that forms the cleavage furrow. So let's write that down. Furrow forms. Perfect. Now, there's one more like thing that happens in telophase, or like it's part of telophase, but it's technically its own little phase, and that is, whoa, you cannot see that. Let's just keep it red because it's technically the same. Cytokinesis. The whole job of cytokinesis, let's move a little over, is to basically separate the materials and organelles and divide the organelles into the two cells. That's literally the whole job of it. So once the daughter cells are formed and cytokinesis is complete, telophase ends and it restarts the whole cell cycle again. So let's just write down cytokinesis is just dividing the cytoplasm and organelles to make sure that each daughter cell has its organelles and it has its cytoplasm and it's ready to function as a new cell. And so that's pretty much it. This is our final process. Our final process, ooh, smiley face. Our final product is 2 daughter cells. So that is mitosis. Let's do a quick review of mitosis. So let's bring, all right, we're gonna make Nogops really big. All right. Whoops, she lost her glasses. We're gonna make her this big. We're gonna draw a new pair of glasses onto Nogops. I'm gonna erase this one. All right, so let's do a review. Interphase. The chromatin is uncondensed and the centrioles form. So uncondensed chromatin uncondenses. That means the chromatin stays uncondensed and the centrioles form. So interphase. Chromatin is uncondensed, centrioles form. In prophase, we got early prophase. In early prophase, the chromatin condenses. So it becomes visible in the nucleus. These centrioles form, and oh my god, the centrioles, they move to the opposite sides of the cell, and then the mitotic spindles, which are these beige lines, they also form, and the nucleus disappears. In late prophase, the nucleolus disappears. In late prophase, the nuclear envelope disappears, so the chromosomes are free roaming. The chromosomes attach to these mitotic spindles through their centromeres, and then these centrioles are going to pull the chromosomes to the middle. And then we go to metaphase. The only thing that happens in metaphase is the centrioles, they line up the chromosomes in the middle. And then we move on to anaphase. In anaphase, the mitotic spindles, they pull and they separate the sister chromatids apart, which leads us to having double the amount of chromosomes in the cell temporarily. And then finally, we go into telophase, where the chromatin condenses again, the nuclear envelope reforms, and then a cleavage furrow forms from the contracting of the microfilaments that are trying to pinch the cell into two. And then one last thing of telophase is cytokinesis occurs, which basically just divides the cytoplasm and the organelles so that the cells can function. The new daughter cells can function. So we go from one cell to 2 daughter cells that are ready to do all their cycles. How does that feel? Are we good on mitosis, Nogops? I think we're good on mitosis. She got this in the bag. We're chilling. So yeah, that was mitosis, and we will do meiosis next, which is a little more complicated, but that is for the next video.